Bismillahirrahmanirrahim. Assalamu alaikum students, how are you? Our next topic, continuously varying traits. Genotype interacts with environment to produce phenotype. Phenotypic expression of the trait has two aspects, qualitative and quantitative. Two aspects of expression of a trait. First one is the qualitative and the second one is the quantitative. Now the comparison of qualitative variation with the quantitative variations. Qualitative differences are large and more obvious, but quantitative variations are small and less striking. इनमें कम difference होता है. Some traits like pea seed shape show discontinuous qualitative variation with two sharply distinct phenotypes, round or the wrinkled. Others like 4 o'clock flower color can have three phenotypes, red, pink and the white. Still others like ABO blood group systems have four qualitative different phenotypes A, B and AB and O. But many traits like height, weight, intelligence and skin color in the humans and grain color in wheat exhibit continuous quantitative variations over a range of many phenotypes. The trait which depends on the quality of the alleles of a gene is called the qualitative trait, and these traits are controlled by two alleles of a single gene. These characters do not vary continuously. Qualitative differences are great and more obvious. One allele is dominant, another is the recessive. They produce only two sharply distinct phenotypes. While the quantitative traits that depends on the quantity of the alleles of more than one gene are called the quantitative traits. These are continuously varying traits. A continuously varying trait is encoded by the alleles of two or more different gene pairs that found at different gene loci. At different loci, all these genes influence the same trait in an additive way. These are the quantitative traits. The quantitative traits are also called the polygenic traits and their genes are called the polygenes. Qualitative, as the name indicates, depends on the quality of the alleles, while the quantitative depend on the quantity of the alleles. Here the quantity, and in the qualitative, the quality of the alleles. And qualitative traits are also called the polygenic traits, in which two or more different gene pairs that are present at the different loci work in a combined manner and effect on the same trait. Mendel focused on traits that showed only two. Here are the differences between qualitative and quantitative traits. Qualitative traits, in this case, phenotypes with discrete and easy to measure values. Individuals can be correctly classified according to the phenotypes. Show Mendelian inheritance, monogene, little environmental effect, and molecular markers are qualitative traits. Examples of these are seed shape, dominant trait, and recessive trait, round and wrinkle, seed color, yellow and green, and flower color, purple and the white, and pod shape, then inflated and constricted, and pod color, green and the yellow. Same is the case in case of the quantitative traits. Individuals cannot be classified by discrete values. Quantitative trait distributions show a continuous range of variation, and phenotypes can take any value. Complex mode of inheritance, as many genes are contributing in a single trait, so they are called the polygene. Inheritance, moderate to great environmental effect. Examples, plant height, yield, disease severity, and grain weight. Is here the graph, that is the plant height 20, 30 and 40, and the percentage of plant 20, minimum value, then is curve shape, and 40 is the highest value. So the plant height is between the 20 to 40. Wheat grain vary in color from white to dark red. This trait shows a continuous spectrum of color variation in the figure 22 to 10. Some grains are white, some are deep red, but most grains have shades in between. Light pink to moderately dark red. Nielsen L. Studied the genetics of wheat grain color when he crossed a true breeding dark red grain plant with a true breeding white grain plant. All F1 grains had light red color, intermediate between two parental shades. It seemed as if it was a case of incomplete dominance. But when F1 grains were grown to mature plants and crossed with each other, F2 grains had exactly 7 shades of color in the ratio of 1 dark red, 6 moderately dark red, 15 red, 20 light red, 15 pink and 6 light pink and 1 white. A cross between a strain of red wheat plant with another strain of white wheat plant. Grain from the F1 was uniformly red, but of shade intermediate between the red and the white of parental generation. And this might suggest incomplete dominance, but when the F1 offsprings were crossed among themselves, the F2 zygote showed 5 different phenotypic classes in a ratio of 1.6.4 into 1 and 0.1. If we symbolize the genes for red with capital letters A, capital A and B, capital B, and their alleles resulting in the lack of pigment production by small a and the small b, we can illustrate the result of this cross as follows. Red parental generation, the red and the white, and the genotypes will be capital A in the homozygous condition, capital B in the homozygous condition. Also in case of white, small a and small b and small b. Now the gametes are formed AB, small a, small b, small a, small b. Next one is the F1 generation. Medium generations are produced and their genotypes are capital A, small a, capital B, small b. In the heterozygous condition, both are present. Now the F1 is crossed with the F1 generation. Then to get F2 generation, F1 generations are offsprings are crossed. Here the table is formed on the above side of the table. Write down the gametes of male, while on the sides, left side, write down the sides of the gametes of female. Here in this case, on the above side, A and B gamete is present. Capital A, small b gamete, then small a, small capital B, then small a and small b. On the later sides, capital A, small capital A, capital B, then capital A, small b, then small a, capital B, then small b and small a. Now combine these gametes in the first box. Capital A and capital A and capital B and capital B, these both are in the homozygous condition, so the plant will be red. In second one, A is homozygous condition while B is in heterozygous condition, so the dark color is produced. And the third one, A is heterozygous condition and B is homozygous condition, so it will be also dark. And the third one, both are in heterozygous condition, so the color of the plant will be in medium. Now the next line in the next box, A is homozygous condition and B is heterozygous condition, so the dark color is produced. And in the next one, A is homozygous condition and B is recessive homozygous condition. So the medium color is produced. And the third one, A is homozygous condition while B is also A is heterozygous condition and B is also in heterozygous condition and medium color is produced. And the same is the case in the third. A is heterozygous condition and B is in the recessive form homozygous condition, so the light color will be produced. And now the third one, first of all, A is heterozygous condition, B is homozygous condition. Now the dark color is produced. And next one, A and B both are heterozygous condition, medium color is produced. And the next one, A is recessive homozygous condition and B is dominant homozygous condition, so the medium color is produced. The next one, small and small a, capital B and small b, color will be the light. And the last one, box A, capital A, small a, capital B, small b, the medium color will produce. And the next one, light color will be produced. And in the next one box, light color also. And in the last one box, white color is produced. All are in the homozygous condition. 1/16 red, 4/16 dark, 6/16 medium, and 4/16 light, and 1/16 is white. If we suppose that each dose of a gene for pigment production increases the depth of the color, we can arrange the F2 phenotype according to the number of genes for the red in the following table. This is a table. Here the genotype, and next one the number of genes for the red color, next is the phenotype, and the next one is the fraction of F2. First देखें, when the capital A, capital A, capital B दोनों होमोजाइगस कंडीशन में है, नंबर ऑफ रेड जीन्स जो है इसके अंदर 4 हैं, तो डार्क रेड प्रोड्यूस होगा और फ्रैक्शन कितनी होगी, एक जो है वो प्लांट प्रोड्यूस हो रहा है रेड कलर का, डार्क रेड कलर का। नेक्स्ट जो है ए जो है होमोजाइगस कंडीशन में है, बी जो है हेटरोजाइगस कंडीशन में है। सेम केस ए हेटरोजाइगस कंडीशन में that it is a pink. Light red, if it has 3 pigment alleles, then it will be light red. 4 alleles of color dose will make red, dark red. 5 alleles of the color dose will produce moderately dark red green. Thus the color phenotypes of the grain is the sum of the individual effect of all 6 alleles. So the 6 alleles are contributing in the color of the wheat grains. Second one, capital A, capital B, small b, capital A, capital A, small b, and capital B, number of red genes 3, so the medium red is produced, and fraction 4. Third one, capital A, small b, next one, small a, capital B, next one, capital A, small a, capital B, small b, then the number of red genes 2, intermediate color is produced, and number of the plant 6. Fourth one, small a, recessive form, homozygous condition, and capital B, small b, and capital A, small b. Here the number of red gene is 1 only, that is 1, and the light red color is produced, and 4/16 fraction. And the last one, small b, 0 number of red genes, so the white color will be produced, and number of plants in the F2 generation is 1. The gene A and B, because contribute red color in this case, therefore they are referred contributing alleles, while that of the genes small a and small b do not contribute red color to the wheat grains, therefore they are called the non-contributing alleles. This is all about the genetics of wheat color. Next example of the quantitative trait is the human skin color. This is also quantitative trait which is controlled by 3 to 6 gene pairs. The greater the number of pigment specifying gene, the darker the skin, and the child can have darker or lighter skin than his parents. And the third one example, human height is more complex polygenic trait. And the perfectly continuous variation in the range of human heights produce a smooth, well-shaped curve. Fig 22. A few people are very tall or very short, but most individuals fall in average or mean value. This trait is controlled by many pairs of genes in different loci. Even multiple alleles may be possible at each locus. More the number of alleles for shortness, the shorter the height will be. Similarly, greater the number of alleles for tallness, the height will be. Environment also has a strong influence on height, intelligence, and the color in the humans. Constant exposure to sun darkens the skin. Poor nutrition prevents achieving genetically determined height. Healthy and encouraging social environment promotes intelligence. This is the human height showed by the graph. The human height is a continuously varying trait. On the y-axis, number of individuals in hundreds of thousands, and on the x-axis, height. And short height is also shown, and the tall height is also shown. And मीडियन की तादाद जो है वो ज़्यादा है एज़ कम्पेयर टू शॉर्ट के और टॉल के. Frequency histogram illustrate variation. A frequency histogram is a simple graph. A horizontal or x-axis indicates the range of different phenotypes of a trait of a population. The vertical or the y-axis determines the number of individuals in percentage in the population. Some people can roll their tongue into a distinct U shape. When turned it out, when they extend it out of their mouth, they are called rollers. This ability is due to a single dominant gene if it is a discontinuous variation inherited in the simple Mendelian fashion. Its frequency diagram forms asymmetric distribution curve with much greater frequency of phenotype at one end than at the other. Human height is a continuously varying trait. If we plot a frequency diagram of the heights of humans in a large population, so many phenotypes are found with categories blending into one another. It forms a smooth bell-shaped normal distribution curve. Measure the heights of a fairly large number of students in your college in centimeters, each to the nearest centimeter. Also note the ability of each student as roller or non-roller. Record your observation in a table like this, and you will form the table: serial number, name of the student, and height in centimeter. Also, they can extend their tongue into U shape or not. They are the rollers or non-rollers. Representing each measurement class as a bar with its height proportional to the number of individuals in each class. Plot the graph. Comparisons of a continuous and discontinuous variations in humans. Here, this is the height, class height in centimeters, and second is the rollers and non-rollers. These are shown in the paragraph in the graphs.